Human Biology, Unit 1, A1, Topic 3, Membranes and Transport Summary. So first of all, just looking at the different components of a cell surface membrane, you've got phospholipid bilayer. So the phosphate heads of the phospholipids are hydrophilic, so they face towards water, either outside of the cell or into the intracellular fluid. And then you've got the fatty acids tails, which are hydrophobic, and they face inwards away from water towards each other. And these are said to be quite fluid because the phospholipids can swap places relative to each other. And then every so often you've got cholesterol, shown here in red, which adds strength and stability to the cell membrane. You also have intrinsic proteins, which are proteins which span the phospholipid bilayer, and these can either be channel proteins or carrier proteins or sometimes enzymes. Then you've got some extrinsic proteins, which are proteins found on the outside or external of the cell membrane. You've also got glycolipids, which can act as recognition sites, for instance, for cholera toxins. And you've got glycoproteins, which are the branching carbohydrate portion of a protein, which acts as a recognition site for chemicals like hormones. So some membrane facts: they are partially permeable and they are made of phospholipids arranged in a bilayer and also proteins. And under an electron microscope, you can see two dark bands which indicate the cell membrane. The fluid mosaic model describes the arrangement and movement of the molecules that make up a cell membrane. The fluid is that phospholipids and proteins are free to move sideways, and the mosaic is that proteins have a scattered arrangement within the bilayer. Phospholipids are molecules that are a constituent of the inner bilayer of biological membranes. They have a polar hydrophilic head, so that likes water, and a non-polar hydrophobic tail, which doesn't like water. Cholesterol is in the cell membranes for fluidity and strength. Glycolipids are phospholipids with carbohydrates attached, and they are for cell signalling and recognition. Intrinsic proteins completely span the phospholipid bilayer. Some act as carriers to transport water-soluble molecules across the membrane. Others act as enzymes. Extrinsic proteins are embedded in the bilayer on one side, but they don't extend through. And they are for cell signalling and cell recognition, or enzymes or receptors. The roles of membranes within cells are for compartmentalization. They separate organelle contents from the cell cytoplasm. For instance, the inner mitochondrial membrane provides a large surface area for aerobic respiration. Some of them contain digestive enzymes, and some of them are where reactions occur. Roles of membranes on the surface of cells are to separate the cell's components from the external environment, to transport materials. They contain enzymes for reactions, has an antigen to recognise cells as their own, receptors for chemical signals. The cell signalling process. One cell releases a messenger molecule like a hormone. This molecule travels to another target cells. For example, it travels in the blood. The messenger molecule is detected by the cell because it binds to a receptor on the target cell's cell membrane. The cell then responds to the messenger molecule by a change in activity. Now looking at methods of transport. So diffusion is the passive net movement of molecules from an area of high concentration to an area of low concentration down a concentration gradient across a partially permeable membrane through the phospholipid bilayer. Molecules that can diffuse through the bilayer are lipid-soluble ones, small and non-polar molecules like oxygen, or very small molecules like water, but only very slowly. Factors affecting the rate of diffusion are temperature, concentration gradient, thickness of exchange surface, and the surface area. A temperature of about 37, a high concentration gradient, a thin exchange surface, and a large surface area are the best conditions. Facilitated diffusion is the passive movement of substances from an area of high concentration to an area of low concentration gradient, down a concentration gradient, across the membrane via carrier or channel proteins. Channel proteins provide corridors that allow a specific molecule or ion to cross the membrane. Carrier proteins are a protein that transports substances across cell membranes, such as glucose, usually large molecules. Factors affecting the rate of facilitated diffusion are the concentration gradient, the temperature, and the number of channel or carrier protein. Exocytosis is a process by which a substance is released from the cell through a vesicle that transports the substance to membrane to let the substance out. So it's where substances exit the cell. Osmosis is the movement of water from an area of high water potential to an area of low water potential down a water potential gradient across the phospholipid bilayer. Pure water has a water potential of 0 kilopascals, and adding solutes to water makes the water potential lower or more negative. Water potential is measured in kilopascals. Now looking at active transport is the active movement of ions. So the active is the only one that's active apart from exocytosis and endocytosis, whereas diffusion, facilitated diffusion or osmosis are all passive. So active transport is the active movement of ions or polar molecules from a low concentration to a high concentration using a carrier protein. This movement is against the concentration gradient and requires energy from ATP and also carrier protein. Active transport facts: molecules move against a concentration gradient. Energy from ATP is required. Specific protein carriers are required to change the shape to move substances from one side of the membrane to the other. Types of molecules that are actively transported by carrier proteins are large polar molecules and ions. Things that can inhibit active transport is anything which inhibits respiration and therefore ATP production, like cyanide, which is a respiratory inhibitor. Endocytosis is the process by which a cell takes materials into the cell by infolding of the cell membrane. Phagocytosis is a type of endocytosis, and it's how white blood cells called phagocytes engulf and digest bacteria.